So I'm Kris. I'm the manager of automation within the solutions team at Covaris. And I want to go back to Eugenio's vision that you mentioned earlier of a 2-day workflow for FFP samples through sequencing. And to achieve that, we pose the question, what if you could go from FFP tissue to sequencing in 2 days with the best quality, the best workflow, and unbeatable economics? This vision is broken into 2 separate workflows, each with their own challenges. FFP extraction is labor intensive. Often, the decarbonization is not automated, even in automation workflows. And this workflow has a very high failure rate, often reported as 20 to 50%. And this can lead to delays, additional costs, or even loss of sample with patients attached to it at the end. Low purity and low yield can compromise the quality of downstream results. For library prep, the workflow can be complicated with many hands-on steps, which introduce risk, as Ryan was discussing earlier. And library prep is a very costly process, so having to repeat it when you have failures is very expensive, very time-consuming. And the last is biases in the fragmentation of the nucleic acids can lead to inconsistent coverage across different GC content genes, which then leads to poor variant calling in your panels. Based on these challenges, we can identify some needs that are present in both. The first is the need for a scalable and flexible automation solution that addresses varying batch sizes and minimizes waste. Second is a seamless solution for automation, which will minimize implementation time, hands-on time, mitigate risks, and improve consistency. And the last is the most important. You need to have reproducible and reliable data that gives good quality and prevents failure and ensures accurate detection. So, as mentioned, LabCorp was one of our early collaborators and early adopters of the Sonication Star, and they've shared valuable feedback and performance metrics since they've implemented this workflow. On the left, we have a chart that goes from early months to beginning of last year, April of 2024. And in the months leading up to their adoption of the Sonication Star, their previous workflow would often drop their full report rates for the OmniSeq Insight report down to 82%, meaning patients weren't getting a full report of their mutations from their cancer, so they might not be put in the right clinical trials. Since the implementation of the Sonication Star at LabCorp, 95% of patients have gotten full reports, meaning more patients are being placed in the correct clinical trials. And using this information, we were able to create a model of the economics associated with this compared to a study with a traditional extraction method. And we mapped out the cost based on the failures from extractions through extraction, library prep, and sequencing, using a model customer running 10,000 samples per year on a NovaSeq 6000 with an S1 flow cell from the TSO 500 panel. And the resulting savings are estimated to be about $1.3 million a year. So, to show the robustness of this vision for a 2-day workflow, we need to look at the 2 separate parts of the workflow. And the first is the extraction. So we engaged in a study where we did DNA extraction using the Sonication Star and compared it to a standard extraction method from another supplier. To achieve this vision, we need to have the Sonication Star deliver high purity and high yield nucleic acids from FFP samples within a single day. The Sonication Star can extract DNA and RNA in parallel, but in this study we only look at DNA. So for this study, we used 4 different FFP cell line samples, and we performed parallel extraction using the Sonication Star and a manual kit from another supplier. The DNA quantity was measured using a fluorescence-based method, and True Extract Smart Solutions maximize the value of every sample by recovering significantly more DNA from each FFP extraction, increasing the chance of success for even the smallest and oldest FFP tissue samples. Purity was assessed by comparing the ratio of absorbance at 260 nanometers and 280 nanometer wavelength, and the optimal range for this is between 1.8 and 2. And the True Extract Smart Solution on Sonication Star ensures optimal DNA purity, which improves the efficiency and consistency of live preparation. Together, this combination of high yield and high purity is an effective way and simple way to reduce costly sample failures, re-extractions, and sequencing reruns. So, the other part of the workflow is the library prep. And I'm going to present this in 2 separate phases, one without amplification and one with amplification. First, we're going to talk about without amplification. So we set out an experiment starting with FFP samples, extracted on a sonication star to get DNA, and then we put those DNA through 4 different library prep workflows. These were all performed manually and PCR free. So the TrueCover workflow is fairly hands-off. It requires only 25 minutes of total hands-on time and just under 3 hours of turnaround time to get from genomic DNA through shearing, end repair, A-tailing, magnetic bead cleanups, and then you're ready for either amplification or library quantification. So in this study, we looked at the first 20 bases from DNA extracted from an FFP lung tissue, looking at the inserts and checking the first bases for A, T, C, and G. In random fragmentation, the individual bases will be present at the same proportions as they are in the genome. With sequence-specific fragmentation, the representation of the specific bases is skewed. Our libraries exhibit a relatively flat, unbiased base distribution, starting from the very first nucleotide. The enzyme-based methods that we're showing here show sequence-specific fragmentation, creating a biased library that does not accurately represent the source DNA. And why does it matter for FFP specifically? This bias can mask low-frequency somatic mutations, which are especially critical when working with heterogeneous FFP tumor samples. Next, we look at the uniformity of coverage across targeted genes with varying GC content. We did whole genome sequencing, but we looked at 504 genes that are found in the TSO500 panel, and we analyzed the differences in coverage of those genes compared to the overall coverage. And what these graphs show is that the other suppliers have much higher coverage at low percent GC and much lower coverage at high percent GC. This can create blind spots in your gene panels, where severe GC bias can lead to systemic undersequencing of key genes and compromise the integrity of the overall panel. TrueCover library prep with AFA fragmentation shows a flatter dose response in relative coverage across the GC percent range. This means that TrueCover ensures comprehensive gene interrogation. You can achieve coverage across your entire panel, including challenging targets. And the impact of this is that by minimizing your GC bias, you're going to be more likely to detect your low frequency mutations and accurate copy number variations in your data. So, as mentioned, we're also going to look at the amplification module that is associated with TrueCover library prep. The following slides will highlight the performance of our amplification kit with a focus on consistency, precision, and recall. We use the sonication star again to extract the DNA. These are from a cell line FFP pellet control, and we performed live preparation with just TrueCover with the amplification module. In parallel, we also worked with gDNA from the same cell line provided by the Cornell Institute. And this genome also has reference data available. That'll be key later. So, the TrueCover amplification workflow is engineered for ultra-low inputs. Our high-fidelity, low-bias polymerase enables robust amplification. And it offers exceptional lab efficiency, requiring only 35 minutes of hands-on time, minimizing technician effort, and freeing up valuable lab resources. And it achieves a rapid turnaround time, just under 4 hours going from gDNA to sequencer-ready libraries. And it just adds these 2 steps to the workflow. So from the extracted DNA from the cell pellet FFP and from the vendor-provided gDNA from the same cell line, we prepared libraries from 1 nanogram, 5 nanogram, and 50 nanograms. The uniformity of coverage across chromosomes was measured relative to the overall coverage, and TrueCover shows consistent uniformity of coverage from as little as 1 nanogram all the way up to 50 nanograms of input DNA. And it's insensitive to the quality of the samples, showing a very similar performance across the FFP-derived DNA and the vendor-provided gDNA for the same cell line. So this enables you to have one robust and reliable workflow that can work across multiple sample types and simplify your protocols and ensure reproducible results. And from the same experiment, we also looked at the true accuracy of using F1 score for these same samples for SNPs and indels. The FFP-derived and genomic DNA samples were analyzed for accuracy of variant calling compared to the genomic reference that I mentioned earlier. For SNPs, we got a score of 0.98 for the F1 score with as little as 5 nanograms of DNA from FFP, matching the performance of the gold standard Coriell sample. And for indel accuracy, the score was greater than 0.92 from the same input, enabling confident calling of insertions and deletions from FFP samples with excellent precision and recall. And this mitigates quality challenges for reliable results. The workflow effectively delivers performance with DNA from FFP, translating that uniformity of coverage I discussed earlier into superior variant calling accuracy. So what does all of this add up to? Earlier I mentioned that there are challenges associated with these workflows, and the challenges are unique for each part of the workflow. So to address the labor-intensive decarbonization, we've fully automated this solution. We start with FFP samples through genomic DNA extracted on the sonication star. Many other solutions do not automate that decarbonization. To address the high failure rates that are reported in the field, we delivered a solution to LabCorp that has a 95% success rate from FFP tissue through the full report of all of their mutations. To address low purity and yield, we have consistently high yield and high purity for optimal sequencing input. To address the complexity of the library prep workflow, we've simplified the workflow and we're working with Hamilton on future automation to reduce the hands-on time and prevent errors. So, when it comes to the costly reagents, rerunning samples is one of the biggest costs, as shown by the model we created before. So, reducing rerun rates will reduce reagent usage and consumable usage, and thus the cost per sample. And we address the fragmentation bias by utilizing a non-biased fragmentation method to deliver uniform coverage and improved variant calling. We address the need for scalability and flexibility by providing a flexible and high-throughput automation solution. And when it comes to seamless automation to achieve quick integration and fully hands-off solutions, we're working on turnkey solutions using the assay ready workstations provided by Hamilton, and we're going to optimize those with Hamilton so that they can be implemented very quickly and effectively in your lab. And most importantly, we deliver high-quality data through improved extraction yield, purity, and coverage uniformity when we use these two workflows together. And thank you for your time. I really appreciate it.